Dear students, I hope you all will be fine. Here I am, Muhammad Jamal Khan, lecturer in botany, University of Education, Lahore. Today we will discuss a topic from one of the core subjects of botany, that is plant systematic anatomy and development. The course code of this subject is BOTN 1112. This is lecture number 26, in which we discuss meristem and its different types that is present in different parts of plant. So here we have the content on the behalf of which this lecture comprises of. Introduction, then we have the characteristics of meristematic tissue, then we discuss the structure of meristematic cells, then we have the classification of meristem, and in the end we have the reference for this lecture. We introduce meristem as these are the highly differentiated tissue made up of immature cells which have not been targeted with any particular function are known as meristems. These are restricted to the regions in the plants which are known as zone of active cell division. So presence of cavity of division is due to meristems. These meristem cells remain embryonic. It means that retain the activity to divide repeatedly. The cells formed by the division of meristems are called derivatives. These cells then undergo differentiation and lose the capacity or capability of division. So they become permanent cells. Here we have the characteristics of meristematic tissue. Firstly, it is an undifferentiated tissue. Cell cycle of a meristem is continuous due to the active state of cell division. The meristematic tissue are composed of immature cells which have not been assigned any proper function. These tissue possess only a primary cell wall which is thin, highly elastic or flexible in nature, and this wall is composed of cellulose. Secondary cell wall is not seen in these tissue. Cells of meristem are really small in size and isodiametric or small cuboidal with dense cytoplasm. The nucleus is large and distinct, but vacuoles are rarely seen. These tissue extremely have active metabolism that give in the exhaustion of the reserve food storage in these cells. Plastids are usually not seen in meristems, but protoplastids, which are colorless, are present. Tightly packed cells result in a compact tissue which result in the absence of intercellular spaces. In these tissue, lack of cellular non-protoplasmic substances that are called as ergastic substances are absent. So now we discuss the structure of meristematic cells. The meristematic cells can be distinguished from unspecialized mature cells because of the variation in the size of the meristematic cells and the ratio between the size of the cell and that of the nucleus varies very greatly in different meristematic cells. So firstly, we have wall of meristematic cells is usually thin, but certain cells in the apical meristem have thick walls. The cells of the vascular cambium have thick radial walls at certain period. The next we have the richer in protoplasm. Meristematic cells are relatively richer in protoplasm. They're usually more isodiametric in shape than mature tissues. The next we have their protoplasm is devoid of reserved material and the plastids are in the proplastic stage. In meristematic cells, mostly the apical meristems, especially among the angiosperm, the vacuoles are very small. They are scattered throughout the protoplast. In pteridophytes and many spermatophytes, some of the cells of the apical meristem contain conspicuous vacuoles. The cells of the vascular cambium are highly vacuolated. In general, larger the meristematic cell, the greater is the degree of vacuolization. On the base of the structure, there are two types of meristematic cells. One is initiating cells, the other one are derivative cells. Those cells retain the meristematic activity throughout the growth of the plant. We call it as initiating cells. And those cells derive from meristems. They mature into different types of cells, and some differentiating cells may retain the potential meristematic power at maturity, like parenchyma, but other loses the potential of meristematic activity permanently. They change into mature cells like sieve tube elements, brachyds, and sclerenchyma. The classification of meristematic tissue is based on the divisional power of plant tissue. So we classify meristem on the basis of 4 criteria under which we discuss over this lecture. These criteria consist of the position of meristem, origin of meristem, on the base of function of meristem, and the division of plane of meristem. So the classification of meristem is based on different criteria as we given below. We classify the meristem on the basis of position, then origin, function, and the plane of division. On the base of position, we have 3 types. One is apical meristem, then we have the lateral meristem, and in them we have intercalary meristem. Next, on the base of the origin, or we can say that on the base of the developmental stages, or on the base of the origin of initial cells, we have 3 types. One is promeristem, that is also called as primordial meristem. Then we have a primary meristem, and then we have secondary meristem. Similarly, we classify the meristem on the base of the function. On the base of function, we have 3 types. One is protoderm, then we have the procambium, and the third one is the ground or fundamental meristem. The last criteria is the plane of division. We have 3 types under the plane of division of meristem. One is mass meristem, the second one is the rib meristem, and in the end we have plate meristem. So these are the different criteria on the behalf of which we classify the meristem. So on the base of position we have three types. First one is apical meristem. So actually these are the cells that are found at the apex of shoots and root. It brings about increase in length. It includes both promeristem as well as primary meristem. In addition to this, sometimes the meristematic tissue is neither apical or nor lateral, but occupies a specific position as based or basal regions of internodes in the grasses, and such meristems are called as intercalary meristem, and these are located in between the mature tissues. The last one we have the lateral meristem. These are situated in the lateral side of the plant body, and these meristem play a role in increase the thickness of plant during secondary growth. So in detail we discuss one by one all these type of meristem on the base of position. So the apical meristem are found at the apices of root and stem. They are called as growing points. So it may be the present at the shoot apex or root apex, and these areas are referred as growing points or growing region. Their size is few millimeters to a few centimeters, depend upon the species of plant. By division, they give rise to a primary permanent tissue and increase the length of the plant in a longitudinal pattern. The next type we have intercalary meristem. These are the separated tissues of apical meristem. These are separated from apex during the growth and found in between the cells of permanent tissues. Their position is different in various plants. Sometimes they are present at the base of the leaf, and in some grasses they are found at the base of internode, and in some cases they are situated below the node. Majorly it present in the leaf base or at the lower internodes of growing stem. Intercalary meristem also increase the length of the plant body. They are short-lived tissues, either disappear soon or convert itself into a permanent tissues. One of the important and major function of intercalary meristem is that they play a role in the production of leaves and flower. So the next type we have lateral meristem. Actually these are cylinders of dividing cells present in the vascular and the cork tissues of the plant. Lateral meristem are present in dicots and gymnosperms, and vascular and cork cambium are the example of lateral meristem. They play an important role in the increase in diameter of stem and root, so they are involved in the secondary growth. These lateral meristem are longitudinally arranged in root and stem, and mostly in two forms. One is fascicular cambium, the second one is phloem or cork cambium. So here we have the image regarding lateral meristem. As you know, the lateral meristem consists of two cambium. One is cork cambium, the other one is vascular cambium. And because these both play a role in increase the girth of or increase the secondary growth of plant. So further we have vascular cambium consists of two types of cambium. One is fascicular cambium, and in between two fascicular cambium there is a interfascicular cambium, and here we have the cog cambium. So next we discuss both the fascicular cambium and cog cambium. So the fascicular cambium simply is the primary cambium that present within the vascular bundle and play a role in the formation of secondary xylem and phloem. While the cog cambium, these are the cells that are present in extraxillary in position during the formation of bark of meristem, and they form three layers basically, and lowermost layer we call it as phloem, which forms the outermost scale bark or phloem. This cog cambium also produce some phloem that are basically the parenchyma cells towards the inner side, but this phloem is not clearly differentiated. It merges with outer part of the non-functional phloem and remaining pre-cycle. So the next types of meristematic tissue is the base of their origin, or based on the development of initial cell. So firstly we have the promeristem, that is also called as primordial meristem or embryonic meristem. In this meristem that is consists of a group of earliest and youngest meristematic cells of growing organ. It is called the early embryonic meristem, from which other advanced meristems are derived, and it is present at the tip of stem and root, and it further divides to form a primary meristem. The primary meristem are those whose cells have been derived directly from the apical initial. It is derived from the promeristem, and it divides in different planes. It is found at the tips of root and stem. The cells of primary meristem form the early structurally and functionally complete plant body. It is always in active state of division, and it gives rise to a permanent structures or tissue. The last one we have secondary meristem. This meristem appears after a certain stage of development of plant organ, that is referred to as secondary meristem. These meristem develop from mature tissues which have already undergone differentiation, and they are always lateral in position. Such meristems develop, you can say, at the time of emergency, like healing of wounds, or to affect secondary growth, or to form cork. Phloem cambium is an example of secondary meristem that occur the actively dividing activity, and it gives rise secondary permanent tissues during secondary growth. This classification of meristematic tissue is based on their function, and in this we have three types. Firstly we have protoderm. It is the outermost layer of the young growing region from which the epidermis develop, so it is called a protoderm. So the next we have procambium that develops into a primary vascular tissues. Actually these are the elongated tapering cells of the meristematic region from which the vascular system develop. These cells, or the cells of procambium, are present very near to the central region. In cross section, each procambium appears as a small group of cells in the ground or fundamental meristem, but in longitudinal section the cells are long and pointed. The next we have fundamental or ground meristem. These are the meristematic tissues that became differentiate into hypodermis, cortex and pith. So these are called as ground meristem. These cells of this region are large, thin-walled, living and isodiametric. In later stages they are differentiated into above mentioned structures such as hypodermis, cortex and pith, endodermis and pericycle. In addition to this, a medullary rays. In the last we have the meristematic tissues that based on the plane of division. So here we have three types. Firstly we have mass meristem. Mass meristem grows by dividing in all planes. So by this it increasing the volume of cells, and it remains present in the endosperm, cortex, pith, help in the formation of embryo, act as packaging tissue, serving the purpose of storage and photosynthesis also. In addition to this, the next we have a rib meristem, that is also called as file meristem. A rib meristem divides anticlinally to the long axis and give rise to longitudinal files or rows. So this growth pattern is seen during the development of cortex and pith. The last type we have plate meristem. In this meristem they divide anticlinally into two planes so that new cells are formed but number of layers does not increase. Thus a plate-like structure is formed. This growth pattern is responsible for the production of uniseriate epidermis and multiseriate flat blade of leaf. So this plate meristem increases the area of epidermis.